Okay, even though this video has started, I am having to try and figure out how to get autofocus to stop doing that, so the blurring. Yeah, it's really obnoxious. All right, I hope that did it. I'm not going to be able to find out until this video is over, so I'll just have to learn the hard way. All right, so nervous system. This is going to be lecture number 2, second part of it. Okay, remember I mentioned before, this time we're going to be going over what's called the synaptic cleft, and we're going to talk about neurotransmitters. I'll get into what those are. You've probably heard of them. That's where you get dopamine, serotonin. You've heard those names, I'm sure. Those are neurotransmitters. Really quick review though, I do want to kind of just make sure we're getting an overall understanding of the whole system. So just a really quick review from the last lecture. As far as the structure, okay, so central nervous system, which is your brain and spinal cord, your peripheral nervous system, which is everything else, and that further subdivides into sensory division, okay. If you'll remember, see if this will be a good color, sensory division is peripheral area going into the central nervous system, okay? I don't think I talked about this last time. But you have several different types of receptors in your body that receive information for different things, okay? So you have baroreceptors for pressure, you have nociceptors for pain, thermoreceptors for temperature and things like that, okay? And so in a sense, from the peripheral area through the nervous system to the central nervous system for interpretation of that, whatever that sense is, okay? So that's the sensory division. Motor division, if you'll remember, is actually the central nervous system going out, okay? I guess I probably shouldn't use the word periphery necessarily. Everything that's outside the central nervous system, okay? Because it could be acting on organs inside your abdominal cavity, your chest, and things like that. So it's not necessarily in the periphery, but outside the central nervous system, probably a better way to look at it. Okay? So the motor division is further subdivided into the somatic motor division, okay? And that is your stuff that you're in control of. So if I want to move my arm, okay, that's the somatic motor division. I'm consciously choosing to do it. Autonomic motor division is outside your control. Okay, so this is automatic. Okay. So, your breathing. Actually, breathing is kind of a funny one because it is both on the autonomic system and the somatic motor division. You breathe automatically, but you can also control it. That's a good discussion to have sometime, okay? But other areas that you don't, okay, like your heart's going to be beating all the time, okay, that's part of the autonomic nervous system, okay? And then within the autonomic motor division, you have the sympathetic division and the parasympathetic division. If you'll remember, sympathetic can be thought of as fight, flight, or freeze, okay? And the parasympathetic, rest, digest. Okay? So, let's see. I mentioned a few things in my last lecture video that I did. Okay, so pupil dilation for one. Okay, if your pupils are activated through the sympathetic division, they're actually going to dilate. Most people, when they think about it, they would think constrict, but dilation is actually when the muscle is being activated, okay? And then parasympathetic, okay, the stuff that's happening under the machine, okay, kind of like just digestion, things that are normally occurring throughout your body, okay? Eventually, I'm going to actually get into these different divisions. There's a lot to be said on each one. They function a bit differently, they're anatomically different, okay? And we'll get into those, okay? And then, if you'll remember, just really quick, action potential, that was kind of the big thing that we talked about last time. All right, resting membrane potential is negative 70 millivolts. You get sodium channels that open, which causes sodium to rush in, okay, until it hits negative 55, and again that's threshold, and that's when all of them open at once, okay? And then you get a massive influx, you get that depolarization on that graph I showed you, okay? And then you'll hit positive 30 millivolts, and then those potassium channels will open, and then it will go back down, hit that hyperpolarization state where it drops below the resting membrane potential, and then go back up to negative 70 millivolts. I don't want to draw all that out because this is just review. If you don't remember it, I encourage you to go back and look at it, okay? It's important to know before we're going on with this. All right, so kind of got a whole neuron here, okay? So again, neuron cell body, you got the nucleus here, there's a myelin sheath, the nodes of Raine-Bier in between. These are your terminals here, axon terminals, okay? These blue dots represent neurotransmitter. Again, that's kind of one of the topics we're going to be discussing here. So, synaptic cleft, I'm going to write this up here just to kind of bring us back into focus when we start getting off topic, because there's so much to talk about with physiology, okay? And we're also going to talk about, of course, neurotransmitters. Neurotransmitters. Hopefully that will stay out of the way. Okay. So the synaptic cleft is actually occurring where one terminal meets with another area, okay? So there's different types, okay? If the terminal of the previous, axon terminal here, okay? If that is hitting a cell body like it is here, that is going to be called, I'll kind of circle this area here. This is going to be called, hopefully I'm not in the way, axosomatic connection, okay? Because the axon's connecting to the soma or the cell body, okay? If it's connected to like out here, that is going to be referred to as axodendritic. Obviously, you see where I'm going with this. Just put axo and then add the part that it goes to. This doesn't happen nearly as often as axodendritic and axosomatic, but you will have sometimes where you will have axonic. Okay, I think it's axonic. Don't quote me on that. It's so hard to remember all of these words, especially when they start getting huge. I'm pretty sure that's right. Axonic connection. Okay. Again, this one doesn't happen nearly as often as these ones. These ones are happening all the time, all over the place. Okay. Another couple important terms I do want to mention, okay, as far as terminology, these are going to be a few things I'm just going to mention really quick because they could be test oriented, like if you have a test that you're studying for, you might need to know these terms. So definitely these three, okay. I totally lost my train of thought. Let me think here for a sec. Okay, that's right. Summations. Okay, so what types of summation are there? See if I got it in my notes here. Again, so many big words. Actually, this word isn't big though. I just don't remember. Okay, so spatial summation is one. Very glad that I choose to have notes there in case I forget things. Okay, and you're also going to have temporal summation. And these are two different ways that a neuron can be stimulated as far as the amount goes. Okay, again, there are different types of summation. Okay, so remember how we talked about the last lecture, how it was the all or none principle. As soon as it hit negative 55, that was it. Okay, it jumped, it went. With this, inside the cell body, when we're trying to build up that charge, that initial charge to get it up to that threshold, there's different things that are inhibiting it, making it go down, and there are things that are making it go up, okay? And there are different mechanisms for those as well, okay? So when you're talking about spatial summation, that is, imagine you have a cell body here, okay? It's a poor looking cell body, an axon, okay? Spatial summation is when you have tons of different neurons connecting, okay? And those are all being added up together. both inhibitory and excitatory, okay? By excitatory, I mean you're causing that depolarization to occur, you're building, it's going to make the firing effect for whatever needs to happen in the body. Inhibitory is the opposite, it's going to be stopping it, okay? And they're needed for different things. So inhibitory actually is necessary, okay? I'll talk a little bit about this more later. But like, for example, if I'm doing a bicep curl, okay, I'm going to need my antagonist muscle, my tricep, to relax a bit for the focus on this, okay? And that happens because of inhibitory type neurotransmitters that will go in there and cause it to not contract. And so it's contracting up here, not back here. There are also diseases that have to do with that, like botulinum. I'll talk about that actually in this video when I get to a different point. It's interesting, okay? So that's spatial summation when it's all connecting to one. Temporal summation is the firing of just one, okay? So we got one. And it's firing multiple times, okay? So it's adding up all the firings from happening from one axon, okay? So remember those words. There's a good chance those will be on a test. Okay. I'll leave those words up there and hope they don't get in the way too much. Okay. So I'm just going to take... Actually, let me check my notes here really quick and just make sure that I'm haven't gotten off track here too much. Okay. So let's take a synapse here, okay? So we're going to take this one right here, and I'm just going to bring it over here, okay? And we'll just imagine this. I'm not going to use the same color, but that's all right. I'm sure you get the point. Let's actually make this a little bit bigger, okay? Okay. And then we'll go down here, okay? There's just one connecting point to another connecting point. Again, this can be on the axon, the cell body, or a dendrite, okay? Obviously, if it's on the cell body or something, this would be a lot longer than this. I'm just doing this to make it more simple for explaining, okay? So what happens, actually, let's do a little bit of anatomy first. So this part right here, this whole area, is the synaptic cleft, okay? This is where all the action is occurring that we're going to be talking about. This little gap is the synaptic cleft. Very, very small. You have to have an electron microscope to even see that cleft, okay? Let's see. So this right here is called the bouton, which is not the type of bouton that you beat people with. It actually means button in French. I think that's how it's pronounced. I'm not sure. It's French. Who knows? French be crazy. And then this right here is the presynaptic terminal, or well, okay. If we're talking about the whole structure, it's going to be the presynaptic terminal. If we're talking about this, just the outside here, we're talking about the presynaptic membrane. Okay. I think you'll probably hear presynaptic membrane more when you're talking about the cleft itself. If you're talking about anatomically at a larger level, you're probably going to hear the presynaptic terminal, okay? And of course, this one would be the postsynaptic membrane. I will be referring to membrane more, at least here, because I'm focusing on the terminal, okay? Let me get a different color here. Okay. I don't know if this is going to work good. I bought a whole bunch of colored markers to kind of bring these things to life a little bit more. Just black and red was a little dull, I thought. So we'll see how it goes and whether we like it or not. So let's imagine that here we had an action potential on a neuron before it went down the axon, okay, from a different neuron. And it came to one of the terminal ends, okay? So what happens is you have that depolarization that's going down the axon terminal, okay, from the sodium channels opening, okay, that we talked about with the last lecture. when it hits the end, you're gonna have, I'm just gonna do 3 here. Obviously, there's who knows how many thousands, millions. I actually don't even know, to be honest, but there's gonna be a lot more than 6, I can tell you that. Okay? So you're gonna have calcium channels. Okay, we've had potassium channels and sodium channels mentioned. But here in the bouton, you have calcium channels that open up, okay? And of course, if there's calcium channels that open, you're gonna have calcium on the outside. A lot of calcium, okay? One of many reasons that calcium is so important, not just for bones. Okay, so when those open, calcium is going to rush into the bouton. Okay, if you wanted to understand how these things work as far as why they go in, that can be, you want to look into osmosis, diffusion, a little bit of biochemistry. I might make a video later having to do with a lot of these things, with these concepts. But as you know, it can be attractive forces, you know, because different ions have positive, negative, just like magnets, okay? So, or it could just be the amount, okay? And that's where we get passive diffusion. So if there's tons of calcium on the outside of the cell, and you know, next to nothing on the inside, and there's a way for that calcium to travel, that calcium is going to run in, okay? So what will happen is calcium, okay, I got some big words I'm going to have to remember here. I do have it in my notes, but I'm trying to learn this myself, so hopefully I got it memorized good. You're going to have a bunch of what are called synaptic vesicles. Okay. And let me get a different color here, just to add some vibrancy. Inside these synaptic vesicles is actually what houses the neurotransmitter. Okay. Vesicles hold the neurotransmitter. There are some neurotransmitters that don't work by this mechanism. Some are actually made in the membrane, like nitric oxide. Also, I think meth actually, or not meth, marijuana. Yeah, marijuana also doesn't have anything to do with these vesicles. Something to do with the membrane. I don't remember that one. I'm not going to even attempt to try and remember. Okay, so that is going to go in. So calcium will come in, and there's actually a protein that connects to these vesicles, and it's called, okay, so we'll just put like right here, connecting here. This protein is synaptotagmin, okay? And synaptotagmin, when calcium binds to it, it's going to cause the vesicle to travel down to the membrane, and it is going to kind of fuse with the presynaptic membrane, okay? I do need to draw a little bit more here, bear with me. So on each one of these vesicles, you kind of have these docking arms, and it's actually called docking when you're referring to this mechanism. Okay, they dock to the presynaptic membrane, and inside, I don't want to use that same color. Inside these proteins, okay, let's do this. Just put a little arrow here. So snare protein. Okay, so it's called a snare protein. Okay. Now, structurally, they are kind of arms like that, but it is a connection of proteins that makes that up, and it is referred to as the snare protein or the snare complex. Sorry, really quick, I'm going to check the phone here, just make sure it's still working, because it's been acting funny. Okay, the snare protein is made up on the part that actually connects with the vesicle, okay? So like the first part here, okay, I'm gonna put this here. This is called synaptobrevin. I think that's an I, don't quote me though, 2. I'm actually not sure what the 2 is. I didn't think to look into that. I should figure that out. Okay, and then the second part of the protein, the part that will actually be doing the docking, okay, is actually 2 proteins, okay. You have syntaxin, and then you have snare 25, okay. So these 3 proteins connected make that docking protein structure, okay. So yeah, I mentioned botulinum before, okay. Botulinum actually attacks, if I remember correctly, the snare 25 protein, okay? And if it can't dock, then the neurotransmitter can't be released into the synaptic cleft, which means you can't reach whatever's trying to be reached to be activated. In this case, it's going to be the muscles, okay? Neurotransmitter can't be released because this protein is being destroyed, which means the muscles can't be activated. Hence, you get muscle flaccidity, muscle flaccid paralysis, okay? The muscles just won't work, okay? It's actually the most deadly toxin in the world. I have heard it said that just a drop of it on a needle is enough to kill 1 million people. I don't know if that's actually true, but that's what I hear. And yet, people use it for Botox to make their faces look even more ugly than they were before, but whatever. So, what's happening here is this, okay, is going to be released. Okay, it's going to release the neurotransmitter across the synaptic cleft. I hope you'll forgive me. I'm going to back up just a little bit here because I want to make sure that we have an understanding. There are proteins. Well, I don't know if I want to draw them because there'll be so much in the way. Okay. I'll just put little lines here, and if I have to erase them later, I will. Obviously, there's a lot more than just 8. But these proteins, called CAM proteins, I want to get this right. Let me look at my notes. So, cell adhesion molecules. Okay, that's what it is. They're a type of proteins that are on each end, and they connect just to hold it together. It's a lot, imagine like Velcro, okay? So, just different types of proteins, serves a different purpose, just to hold these 2 ends together, okay? So, neurotransmitter is going to be released in the synaptic cleft, okay? And on the other side, do this, you're going to have different types of channels depending upon what's needed and what's going on. And actually, probably ought to. Okay, so what's going to happen is your neurotransmitter is going to come down and it's going to connect, okay, to these channels. And then the connecting of the neurotransmitter to the channel will open this. This is where it starts getting complicated because there's so many different types and different ways that things are activated, and we really can't cover them all. I'm going to cover some of the main ones, and which will kind of give a good overview and of all the different ways that they work. It'll be like a broader classifying of it, and then getting down to the nitty gritty, there's little differences in each of those classes. Okay, so side note, you can get a lot of things, different types of drugs that we have, like serotonin specific reuptake inhibitors, anti-anxiety, anti-depression meds, a lot of things that are really hot on the market today. What those actually do through different types of mechanisms is that keeps the neurotransmitter inside the synaptic cleft. Because normally, under normal function, the neurotransmitter is going to be taken out, different things can happen to it, different mechanisms, but it's not going to stay in the synaptic cleft. And those types of drugs will actually keep the neurotransmitter in that cleft just to keep the firing occurring for, to activate whatever needs to be activated, for your mood, the pleasure system in your brain, different things like that. Okay, there's a lot of debate on how safe that actually is, because a lot of this information is really unknown as far as long-term effects. We haven't had these drugs for terribly long. So something to consider if you think about getting on them. I have actually taken serotonin-specific reuptake inhibitors, which keeps the serotonin there, and that to deal with anxiety, social anxiety, and things like that. And I can say it works miracles. It really does work. But I stopped. I do kind of worry about what that could do in the long term. We don't really know. So if you decide to do that, even if your doctor really tells you it's a good idea, I think it's a gamble in my opinion. And because I'm on YouTube, I'm going to say anything medical, don't listen to me without talking to your doctor, just in case. Okay. So so this is what we would call a chemical synapse, okay? There are also electrical synapses, which are a bit different. It's kind of funny, back in the day, everybody thought that all these synapses that were happening were all electrical, and then studies found out that it was chemical. But now we're finding out recently that a lot of things actually are electrical, but they're the exception. Except for when it comes to a couple things, like cardiac muscle and the uterus, actually. Excuse me. So let's say you have two cells. I'm not going to draw the whole cell, but they're connected, or they're really close together. 2 nanometers, actually, very close, okay? And inside each one will have these 6 proteins, okay? Yeah, let's do this. Imagine that that's 6, okay? And these are called connexin proteins. Not the most thought out word ever. They connect, so let's just call them connexin protein. All right, and they actually will connect, and they form a channel from one cell to the next. All right. So if we're trying to, all right. So in the case of cardiac muscle, imagine these are cardiac cells. You're going to get an influx of sodium to cause the contraction to occur. Okay. So if sodium rushes into this cell, it has the power to also flow from this cell into this cell. And that's why your heart, when it contracts, starts up at the pacemaker cells I have a video on the heart if you want to look into that a little bit more. But there are pacemaker cells that kind of set the tone for the beat, and it will start there. And because of these connecting proteins, those ions will travel through, and the whole heart will just kind of contract, clear down and then down to the ventricles, and back up the ventricles to pump that blood out. Okay. The uterus also does the same thing when you're giving birth. Okay. Functions a little bit different because oxytocin is at work in that one. Oxytocin is a hormone. It comes from the posterior pituitary gland. It's actually behind the eye, and it will cause the uterus to undergo that same thing, and and all the cells are connected. So that's a different type of synapse. Let's kind of start this over. All right, so I want to kind of delve into the different methods here for how the neurotransmitter activates the other side. And when I'm doing it, you'll see there's a lot of areas that have to do with biochemistry. And again, I'm going to actually try to not get too caught up in that. It's not really the area we're talking about. Physiology is a lot more cause and effect type stuff. Okay, you do one structure does this, that acts upon another structure. Okay. Okay, gonna look through my notes here really quick and make sure I'm doing okay. Okay. So, a good area to start, okay. One main neurotransmitter that you will hear about a lot is acetylcholine. Okay. I don't know for a fact if this is the most used, but with how much I read about it and it's always used, I think it's safe to assume that it's probably the most used. Okay, acetylcholine, and we always abbreviate it like this. Okay, it's just a lot easier to write. Acetylcholine is actually the combining of acetic acid and choline. Okay. So there are two different types of receptors. Okay. You have nicotinic. They call it nicotinic because it is activated by nicotine, actually. Okay. And then there's also another type of receptor called a muscarinic. Muscarinic. I'm actually not sure which way it's pronounced, but I say muscarinic. And that is activated by a type of chemical that comes from a mushroom, actually, and I can't remember at the top of my head what that mushroom is. I'm going to say it's a safe bet to assume it's probably something to do with MUSC there, okay? So let's draw again, so this is the postsynaptic membrane, okay? And then if we have, and this is the presynaptic membrane. That's not in the way too much. And then we'll assume that neurotransmitter has been released through the synaptic vesicles that connected through those docking proteins, and it's filling this synaptic cleft. Okay? So the nicotinic is also called a ligand-gated channel, okay? So these different things, we would also call these ligands, okay? Ligands. A ligand, basically a molecule that attaches to a receptor protein is a ligand, okay? In this case, it happens to be a neurotransmitter, okay? So in this case, you can call it a ligand or a neurotransmitter, okay? So we're gonna make a really big gate here, okay? And then this, let's do, here, I'm going to do boom, and a little pocket here, and a little pocket here, okay. Then I'm going to try and make this pretty a little bit, fill this in with some color. Okay, so the neurotransmitter is going to go down, and as soon as it takes two, okay, so one neurotransmitter attach here, another one attaches there. It is going to directly open up the specific channel that it attaches to. So it's a receptor protein, but it's also a ion channel, okay, or a protein channel. Okay, so this becomes activated, it opens up, and then sodium is able to rush in, or whatever ion is needed. Okay, there are some different cases. But generally, it's going to be sodium. Okay, so you got some sodium. Na plus, okay, and it's gonna rush through the channel, okay, and then that is going to cause electrical charge to start to build, whether that's depolarization or hyperpolarization, okay. All right, so that one's a really easy one. That one's a very basic, okay? Gets a little bit more complicated when we start looking at the muscarinic, okay? I'm actually gonna leave that one up there and see if I can just build another one down here.